Good afternoon, and thanks for everyone for coming. And first off, I want to thank Amber, and like any other speaker, it's really because of Amber that I'm here to share some of the work at Boston Children's. But also, I want to thank her to put me my talk, which is focusing on gene therapy after Stephen Gray, the expert in AAV gene therapy, not directly behind him, so it takes up some pressure. But basically, what I wanted to share with you today is some of the targeted approach that we think about maybe the next generation of the gene therapy, if you will. And I'm Henry Lee. I'm a senior scientist at Boston Children's Hospital. My mentor, Dr. Alexander Rothenberg, is sitting at the back. And I live in two worlds. One is the Boston Children's, but also we co-founded Galibra Neuroscience, which hopefully will translate some of the work that we are doing in the lab and to the clinic. And it now doesn't work. Okay. So already you have the disclosure. So the objective for today, I would like to talk a little bit about the preclinical work that we think about a gene therapy, a targeted approach for SLC6A1 related disorders. But also, for the second part, or a smaller part of the talk, we'll talk about some scalable path for this gene therapy. Right, so I have good news and bad news to start with. The good news is that gene replacement therapy has been getting good traction in the recent years. On the left, you can see a very simple search of the term gene replacement therapy in the PubMed. So it's a publicly available database that you can actually see that the number of gene replacement therapy, this term has been gaining so much attraction or attention in the recent years. But if you zoom into those 500 or so publications and thinking about where they are focusing on, the targeted for the central nervous system, CNS, is actually not too much. So you can about 6% of those publications. So the question is why? We are still having a lot of challenge in this CNS-oriented therapy. So when we zoom in, so as Dr. Gray has talked about, there are approved AAV gene therapy already, and this is pretty much the summary starting from maybe a decade ago from Europe. And here is basically the whole list of the approved AAV therapy. You can actually see that there are only one, the AAV9, that the Zolgensma, that is actually targeting CNS related. And all the, the AADC deficiency, that's brain related. And so that also started from Europe and in 2022, and it's got approved this year. So there actually has been a lot of challenge for brain-targeted AAV gene approach. So we want to think about what is the reason and how we're going to target those challenges. I think it wants you to take a better look at this table. Maybe you can help me to click to the next one. That might be easier. Yeah, so the first thing we think about, there are several challenges. One is that the SLC6A1 or gene of interest today is actually brain-wide. So we want to, when we think about gene replacement, we want to pretty much cover the whole brain. And on the left is a in situ hybridization. We look at the transcripts of SLC6A1 across the brain. So you can actually see that there are region specificity, but pretty much SLC6A1 is across the brain expression. So when we think about replacement, how are we going to do that? So Dr. Gray talked about the AAV adeno-associated virus, and which is a prevalently used vector for delivering a gene. But it has a limitation. So if you look at the right in those brain on the top, the AAV9, that's the and the panel C, the top, the second graph, pretty much is black. But we actually see that there are AAV expression there, AAV9, but it's just not very much. In the lab, we are actually have that technology now. You can see that by changing the capsid of this AAV, we are able, in the lab, we're able to express the transgene expression a lot more. So you can see that the gradation of that change of the green signal is much higher. But unfortunately, this AAV PHBD-EB is not usable in clinic because it is not passing blood-brain barrier in patients. So it's worked in mouse beautifully. So we need to find some vehicle that would work for this translation. If you go to the next one, please. the next challenge is that, okay, if we have a vector that can go to the brain, the next question is where the expression of SLC6A1 is. So again, going back to this single-cell RNA sequencing, where we look at single-cell expression of SLC6A1, on top, which is basically from an adult brain, and we ground up and separate into single cells, and then align them to ask the question, what is the expression profile of SLC6A1? So you can see on the left, there are a lot of red bars. Those are interneurons, so that's a major inhibitory neuron in the brain. And you'll also see that on the further right, there are some little purple bar, that's astrocytes. So those are actually the two major cell types that we know that SLC6A1 express. You can see pretty much all the other cell types are silenced. In the panel B, there's another way to look at this. You can see SLC6A1, the bar next to the SLC6A1 label, you see that a lot of warm color, the yellow and white color. So that tells you the expression, again, is in the GABAergic, again, inhibitory neuron. But then pretty much silence expression in the other types, glutamatergic. There are also a little bit of SLC6A1 in the GFAP and the oligodendrocytes. So those are the glial cells. So again, this is we're talking about the brain cell type specificity of SLC6A1. So when it comes to replacement, we want to do that. If you go to the next one. So how are we going to address these cell specificity measures? So we are thinking, we're going back to the mother nature and ask and get the answer. All the genes that we have in the brain, in the body, actually it's inherited from our parents, and all the cells have the same type of genetic material. But in the brain cell or specific cell types, there are differential gene expression, and because regulated by this promoter sequence, or what we call the regulatory sequence. So we just arbitrarily take, because of the limitation of the AAV virus, we can put about 2,000 base pair or 2,000 alphabets of this genetic material into this AAV, and using that as a genetic instructions to drive the SLC6A1. So this is kind of like the model that we are trying to develop, and we think that using this native promoter approach will restrict the expression of SLC6A1 mediated by the AAV. Next, please. And the other question that we are thinking about is that we also heard about today that SLC6A1 is an autosomal dominant disease. So that means one mutated copy and one good copy of the gene. So from Katie Kang's work, we know that SLC6A1 mutant actually stuck in the endoplasmic reticulum. So the protein product of the variant actually is not playing a functional role, but actually it's playing some physiological impact inside the cell. So when it comes to gene replacement, we think about whether this ER trafficking would actually be hindered by the variant. So if that's the case, then do we need to address that? So we also we heard previous speakers about antisense oligo and also 4-phenylbutyrate. We think that there might be some levels both at the transcripts and protein level that we can target those variant protein. And that might be an opportunity for a combinatorial drug and gene therapy approach. Next, please. So when we think about another aspect is the neural development. So SLC6A1 encode GABA transporter, GET1. By now you should know that. And this is a very simplified cartoon and showing what is the physiology of this GET1 mutation. So basically, it's the impairment of the reuptake of GABA from the synaptic site. So there are two things that we can think about besides the SLC6A11 that we talked about in the past talk. There are two things that we think that this impact of GAT1 mutation might have. One is that because of the dwelling of the GABA in the synaptic site, the receptor might internalize. So that's the purple part. But also that the other part might be the GABA synthesis. So we know that GABA needs to be constantly synthesized and degrade. And in terms of the GAT1 mutation, there is impairment of the GABA reuptake. So imagine in the presynaptic terminal where you have a reduction of GABA because of the reuptake is impaired, there might be a compensatory upregulation of the GAT, which is the enzyme that synthesizes GABA, the glutamate acid decarboxylase. So If that's the case, then when we put back the gene and then to renormalize this system, can these compensatory changes be reversed? So that's the question that we might want to address. So next, please. So we actually see some evidence at the molecular level in mice. So this is a collaboration with Jackson Lab. They made a mouse, engineer a stop codon into the SLC6A1 intron. So basically, it's a knockout model. But we also enable what we call the Cre-Lox recombination, take out that stop codon to restore the gene. On demand. So we are just starting this work and characterizing this mouse. And in the cortex, we do find that the GABA synthesizing enzyme, GAT67, actually is increased, so as shown in the middle panel. But on the other hand, the gamma-2 subunit of GABA receptor, which is the obligatory subunit in the synaptic GABA receptor, is actually showing not much of a reduction. Maybe there's a trend. So hints already showing that there seems to be some compensatory changes that seems to be aligned with our hypothesis. The question is then, when we turn on the gene, mimicking that gene restoration scenario, can these changes be restored? So this is a scenario in cortex. When we look at in the other brain regions, next please. For example, this is in the cerebellum tissues from the same mouse, but we don't see that changes at baseline. So maybe a brain-targeted approach or some brain region-specific monitoring will be needed when we look at this gene replacement therapy. Next, please. So to summarize this part, in the preclinical level, we think there are several things that we might need to address. One is that SLC6A1 is brain-wide expression, but it shows a cell-specific expression. So we might need a very good blood-brain barrier penetrable virus, maybe next-gen capsid, for this brain-wide delivery. We also might need some regulatory element that might restrict cell-specific expression. But thanks to Dr. Gray's experiment and also have a very good insights on maybe this might not be necessary. So that can be a good news for the downstream development of this approach. The second point is that from Kedikan's work, we know that the variant of the protein might be having some maybe some endoplasmic reticulum stress, ER stress. And those kind of unwanted interference at the protein level might need to be addressed. So we might think about ASO or 4-phenylbutyrate as a combinatorial approach in addition to the gene replacement. And finally, is the reversibility. We might need to have a model that recapitulate the disease conditions at the beginning, but also we can do a very controlled restoration approach to see if these conditions can be reversible. So the ongoing work with the lock-stop strategy in the mouse will tell us that kind of information. Next, please. But this is just SLC6A1 for the clinical path. We think that because it's one of the rare disorders, it might be facing a lot of hurdle in terms of downstream development. So we want to zoom out and then just to see if we can make it into a platform approach. The approach that we take is combining a group of GABA homeostasis disorders. So this is a cartoon not only focusing on SLC6A1, but now we're looking at many proteins focusing on GABA homeostasis, the recycling and the GABA receptor, and also degradation. So there are about 23 of those genes that are related to that signaling and homeostasis. And interestingly, many of these disorders are actually sharing a common phenotypes in patients in the clinical populations. For example, epilepsy, we have heard about it, and developmental delay and autism. And so it's not unique to SLC6A1. We think that some of the shareable underlying physiology might give us some insights, and when also thinking about the gene replacement. So the treatment options of these homeostasis disorders are mostly asymptomatic. So gene replacement is now gaining traction. It might be a good timing for this emerging technology to become a real thing. And good thing is that many of these genes are actually small enough to fit into the AAV vector. So we think that that could be a practical approach to go in this targeting this big class of GABA homeostasis. Next, please. So my journey actually started from a related disorder called succinic semialdehyde dehydrogenase deficiency, SSADHD. And this is basically a brief timeline of that disorder, if you will. It's a cousin of SLC6A1. The disorder, I will tell you in a little bit. But basically, we made this lockstop mouse model in about 2020. And within from 2020 to now, we're already getting the mouse, and then we show the reversibility of that conditions. And we also have a potential clinical product that can go to human trial. And we ambitiously and optimistically think that we might be able to do a first in human within 3 years. And last year, we formed Galibra Neuroscience that we think that academic research can only do so much, and we will need some of the downstream vessel to really take that to the endpoint, to the finishing line. Next, please. So what is SSADH? Very briefly, it's an enzyme that degrades GABA. So we heard about succinate in the Krebs cycle, in the TCA cycle, in the citric cycle earlier. And this is exactly the disease that without SSADH, GABA is accumulated, as well as a metabolite, gamma hydroxybutyrate. And the red line in the bottoms here is a lockstop mouse that without treatment, they all died at 3 weeks. And, but with our AAV approach, we're able to rescue some of these animals. So there should be 2 videos. If you can, next, if you can show, well, so somehow the middle one is missing. It's supposed to show, imagine there are 2 mice. One is really sick, the other is not. So in here, this is after treatment. And so there are 3 mice. One of them is a homozygous. It's a knockout mouse, but being treated. And I think the bottom line is that you are not able to distinguish this. So in other words, we are able to use AAV to rescue the phenotype to a complete manner. Next. Oh, now, okay. So what I said is true, that this mouse is really sick, and they are showing a seizure and ataxic conditions. So I think we can move to the next one. I'm mindful of the time that I'm blocking everyone before the break. Next, please. So, okay, maybe next. Yeah. So this is where we are now. So I'm grateful for my mentor, Dr. Rottenberg, and we found this startup company. And we're also very grateful for Amber's participation. She has been really helping us to develop on the business side because of her network, and really connecting us and learning a lot along the way. And we also have a strong support for the SSADH Association and 2 experts, one from the clinical side and also the other one from the AAV gene therapy space. So next, please. So this is final acknowledgement. Just want to acknowledge everyone from the Rottenberg Lab for their help, for their really diligent work on this project. And I have not had the time to talk about some of the parallel iPSC-induced pluripotent stem cell patient neurons from the Dr. Mustafa Sahin's lab at Boston Children's. And also Dr. Tim Yu, so we also collaborate a little bit and thinking about the ASO strategy. And finally, most importantly, I want to thank Amber for her support and really learning a lot along the way, and also the funding from Cure Epilepsy Partnership Award. Thank you for your attention.